Fergelot, sorry, about killer whales.
Okay, good morning to all. So today I will present you my work on the Crozet killer whale stereotype calls and their variation across the social units. So we are listening to cetaceans because they are highly vocal animals. In fact, they are using sounds for a lot of purpose, such as communication or foraging, reproduction. And the emergence of new technologies allow us to do some passive acoustic monitoring, which is a non-invasive method. And it can provide us some new information on cetaceans population, such as understanding the link between the acoustic behavior and the ecology of the populations. So for a reminder about the killer whales, you may know, but they are part of the cetacean order, and more specifically the Odontocete suborder. They are present in all oceans over the world, and they usually live in matrilines upon the supervision of a matriarch. And here there are the different ecotypes. You can find that all over the world. An ecotype is a type of killer whales that exhibit a specific behavior with the foraging, the phenotypic traits, and also the acoustic dialect. So we are listening to killer whales because we use it as an identification, as an indicator of the identity of the groups of killer whales we have. We already know few things about them, like fish eater killer whales tend to exhibit a higher vocal activity when they are feeding than the ones that are fed on marine mammals. We also know that a fluid social structure from the killer whales tend to have a more diverse vocal dialect. So for the killer whales, they are able to emit 3 different types of signals. There are the echolocation clicks, the whistle, and that they're used for the close range communication. And we also have the pulse calls or stereotype calls. So this is the one we are going to focus on. They are the most frequently studied in the world because they are also the most recorded ones. Since they're really stereotyped, it's easy to categorize them into call types. And so with those call types, we had their vocal repertoire. So for the killer whales from the Crozet Archipelago, we only have one study on their vocal behavior 35 years ago. And in Crozet Archipelago, we can find out 2 different ecotypes of killer whales. We have the common regular Crozet killer whales, which is the most known, and the type D, which is less known because more rare to see. So for the regular type killer whales, they're studied for over 40 years now with photo identification and genetics. They exhibit a generalist foraging behavior. That means they are feeding on marine mammals, penguins, fish, which is uncommon for killer whales that are usually a more specific foraging behavior. They are known for the depredation of Patagonian toothfish on longline fishery. So for those who may not know, depredation is an opportunistic behavior of the marine mammals that fed directly on the fishing lines or nets. And so due to that, and the illegal fishery in the area in the 80s, the matrilines has disappeared, a lot of them, and so now it resulted in a real social structure of the population. That means they are not living in matrilines anymore. So here we just focused on how the specific ecology of Crozet killer whales influence their acoustic behavior by asking ourselves how the foraging ecology impact the vocal behavior and how the social context of when they were recorded influenced the call type variations. So for the data acquisition, we have put in place 2 different methods of data acquisition. So first we used a hydrophone from the Ligurian instrument band with a sampling rate of 96 kHz, which is enough to listen to the killer whales. We have put the hydrophone during the long line hauling phase of the lines because it's when the killer whales are doing the depredation, and so there are really high vocal activity. And in parallel, we have put in place photo identification method just realized by Erwan Ojan, and who had take picture of all the dorsal fins of the individuals around the boat in order to identify them. So this campaign resulted in 19 records in total with duration ranging from 30 minutes to 2 hours. So we had in total 24 hours and 11 minutes of record to proceed. For the photo identification part, we had identified all the individuals we had during the records, and each individual was associated with his belonging social units, because we couldn't keep the individuals because it's too complicated to know which one is talking. So we had to choose to just keep the social units, and we had identified over 9 social units. So those social units, they were mostly seen together. They were just only one time we had one social unit, but otherwise we always have different social units in the same time in the area. So we choose to let them like that, and to make like a group combination that I'm going to talk about from now as a situation. So for the acoustical part, I have proceeded to manual identification of all the signals in all the records. I have found out over 2,400 pulse codes in the records. And in order to make a catalogue or a repertoire over them, I had made a high quality selection of the pulse codes. That means I had manually annotated each pulse code with a mark from 0 to 3. 0 was a bad quality signal, and 3 a good quality signal. And so for my catalog, I just written the index 2 and index 3 signals. And so I had written almost 600 calls for the catalog. And so for the catalog, here are some of the types we could have. I had made an oral and a visual examination for them. And so I was just ordering them with that. And so it ended up in I found like 21 pulse codes types in total, which is huge, because with our dataset of 600 calls is a lot of different call types. In comparison to Icelandic killer whales that are really, really known, they have found like 52 call types, but with a dataset over 9,000 pulse codes. So we could already say like that Crozet killer whales have a really diverse repertoire of pulse codes. And also we have a high vocal activity of the killer whales, because there is almost like 25 calls per hour, which is huge compared to the Icelandic killer whale with 13 calls per hour. And after that, I was looking at the relationship between the call types and the different situations we had. So I have found that the more social unit we had in the area, the more different call type we had recorded. And when we take a deeper look at it, we could see like this is a similarity index of first with the social units, and then with the different call type we had, depending on each situation. And so we can see like between the situation 1 and 2, there is a lot of shared social units, like there is almost all the same individuals in the area. And that way we have a lot of shared call types in the same time. But an interesting situation is the situation 5, because there is no shared social unit with any other situation, but still we can see that there is shared call types in all our records, particularly with the situation 2 and 6 that shared like more than the half of their call types. So it's an indicator that there is a common repertoire with shared and specific call types over this population. So for the message to bring home, the Crozet killer whales, they have a high vocal activity and a really diverse repertoire. Yeah, they have a large quantity of call types despite a limited data set. And we could, like, if we put it back in the context of the depredation on Patagonian toothfish that I said before, killer whales tend to exhibit a more higher vocal activity when they're feeding on fish. So it makes sense that our killer whales are exhibiting the same behavior. And they are also mostly shared pulse code types across the population. So it means there is a presence of a large common repertoire across this population. with a high diversity of shared postcodes. And it could be due to the recent restructuration of the population, because as I said, this is a really fluid social structure and not living in matrilines anymore. And so for the perspective, some new recording campaigns could provide information on first the link between the acoustic behavior of the specific ecology of those killer whales and their acoustic behavior. We could also have information on different vocal behavior depending on the prey, because as I told, Crozet killer whales have generalist foraging habits. And we could also see if there is an evolution over the time, and if their social units had more specific call types from their own and not shared anymore. Here are some of the references, and thank you for your attention.
All right, thank you. Do we have any questions? No one? Okay, I'll go first then. I think I have two questions. The first one is, so I think you already answered a little bit that question, but do you have any opportunity to confirm that some call types were in fact produced by different individuals, so that they could be like really a shared repertoire, while some could be produced by only one, and then it's probably just call variation? You know what I mean?
No, I have perhaps I have the I have some more. Yeah, it's over here.
Yeah.
It's all the call type depending on all the situations. So we can see that the call type occurring in different situations. And so it's probable that they are shared across all the population and not only just for one social unit.
All right, great. That is exactly what I was expecting to see. And the last slide you talk about, sorry, is there another question? Okay, good. The last slide you talk about ecology. Yeah. What do you mean by ecology? Like finding the link between ecology and some call?
Because of the specific ecology of the killer whales over there, because they're like, as I was presented in before, they have fluid social structures since the disappearance of the matrilines over there, because of the illegal fishery. So this is really uncommon. And also the second thing that is uncommon, it's their generalist feeding behavior, because it's more rare than usually they have, like, I mean, usually when they are feeding on fish, they feed on fish. That's it. They're not gonna search for anything else. But those ones, they are fishing on absolutely everything, which is really uncommon.
So the goal would be to compare with another population, maybe. So
yeah, we already have compared with some population, and we definitely don't have the same call types, I would say. But we have the kind of the same behavior, like when I was saying, like when killer whales are foraging on fish, that they are showing a more activity, vocal activity, and that's what we found over here. So.
All right. Okay, do we have any yes?
Thank you very much. I'm not sure I understood what's the depredation phenomena. Could you explain it again?
Oh yeah, okay. For the depredation, it's just, come on. It's an opportunistic behavior of the killer whales. In fact, like the fish are on the lines because of the fisheries, and so the killer whales, they are just coming and pick up the fish on the line directly, so they don't need to forage anymore. They don't make any efforts. They just pick it and go, and so they don't need to be silent at all, because like the prey is already dead, so they don't care.
Thank you very much.
Thank you. Yes, I think it's maybe a very naive question, but I'm not sure. But I will ask anymore. Anyway, I'm surprised by your conclusion about, there is with your number of calls, there is 21 call type, and your conclusion is this is a very diverse there is a very wide diversity of pulse type, because there is a big little part of coal, and in another population there is I don't really understand how can you conclude on the diversity of the pulse coal due to your number of samplings. It is because maybe if you have more pulse coal, it's due to individualities or maybe something like that, or I'm not very aware of.
I'm not sure I get it, but
it's just you know the line between number of calls and diversity, and your number of pulse calls. You're just a slide before. No, this is one. Yeah, data set. No, this. Yes, this one. Yeah.
Okay.
The data set. 500 of pulse code.
Yeah.
And 21 call types. And 52 pulse codes and more than 9,000 pulse codes. And your conclusion is, because there is far less pulse codes, there is probably far more diversity possible. And you state that there is, it's not even a potentially, it's there is very diverse. And I don't
because okay, it's just because I compared in the literature what we had already, because there is some populations that are well known, such as the Icelandic, but also the British Columbia population, and they exhibit like I don't remember exactly the number of call type they have, but compares to our number of call type we had found in this little sample, it was I mean like we had a huge.
It's the ratio. Like the ratio here is better than the one before. Yeah, could be.
Yeah,
could be that you.
That's why I was saying like we should have new records campaign to get more information, because this is just the beginning, and so I can't conclude for real, but I could already say that.
Good. Thank you. Is it a good one? All right. Next talk about bottlenose dolphins. Let's go.
Okay, thank you. Hello everyone, I am Alice Mengarelli from the University of Turin, and today I present you very first results of my PhD, that is about visual and acoustic approach for the conservation of the Mediterranean bottlenose dolphin. So first of all, yeah, I mean in the PowerPoint there was a bit of animation, but we are in the Mediterranean Sea, specifically in the Ligurian Sea, that is part of the Pelago Sanctuary for the Mediterranean mammals. In this area, there is this long-term project called Delfini Metropolitani of the ONLUS in Italy, Fondazione Acquari di Genova, that has the aim of monitoring through the traditional method of photo-identification the geographic unit in this area of common bottlenose dolphins. Here, in fact, this species is regularly found in the shelf waters of the sanctuary. However, the photo identification, as you may know, is a traditional method, and so is time and energy consuming. So, for a few years now, bioacoustic data are also being collecting on free-ranking individuals in this area, thanks to the collaboration with University of Turin. You may know common bottlenose dolphins as a very complex acoustic repertoire. We find, for example, whistle that are frequency modulated narrowband sounds, and a specific type of whistle is represented by the signature whistles. These whistles, in fact, are individual frequency patterns, so This means that each individual has its own, and this signal broadcasts the identity of the individual thanks to the modulation pattern of the signal. And they are actually stable for decades, so for many years, and learn during the first year of life, and they act as a social glue. So they are commonly produced when individuals are isolated, or also to coordinate individuals in the group. So we can identify them in the recordings using a method called CGIT method, that is a temporal method that I'm not describing here in detail. So even though the common bottlenose dolphin is one of the most studied species worldwide, in this area its acoustic behavior remained poorly explored. In fact, we only have one study of 4 years ago that is focused mainly on signature whistle only. So we had several goals. First, to try to define a catalogue of the signature whistles and variance whistle that are non-signature whistles, so whistles that not respect the CGIT method, so without a stereotype frequency contour. And then we also investigated what factors influence the occurrence of signature whistle and of specific categories of whistles. And we also tried to investigate if the number of inflection points was affected by the presence of trollers and calves. So the inflection point is the change in the slope in the contour of the whistle from negative to positive, or from positive to negative. And to understand what trollers mean, we can see the next slides. So to do so, we used a data set collected by here present Bianca Bonelli for her master thesis in 2023 in an area between Genova and La Spezia. And so dolphins were found visually scanning the surface of the sea, but also searching for the trawlers, so for the fishing vessels, because here it's very common that dolphins follow these trawlers where they can find concentrated sources of food, and so there is this strict association also in place. And so the hydrophone was lowered into the sea from an inflatable boat. So the recordings were subjected to acoustic annotation in Pratt, where we identify the signature whistle using CGIT method, and we also classify the variance whistle, the frequency contour of the variant whistles, and then we extracted the acoustic features with PAMGARD and also the frequency contour of the signature whistle using Python. So at this point we did some statistics in R. We ran some models. First, a binomial one to investigate the effect of group size, boat presence in relation to the calves, trawlers presence in relation to the calves on the signature whistle occurrence. So boat presence is different from the trawler's presence because it refers to the presence of any type of boat within 500 meters from our sighting positions. Instead, the trawler's presence refers specifically to the fishing vessel that dolphins follow. Then we ran a multinomial model to investigate the effect of group size, calves, and trawlers on the occurrence of specific categories. And finally, we ran a post-1 binomial model to investigate the effect of trawlers' presence in relation to the calves on the number of inflection points contained into the whistle. What we have. So from almost 6 hours of recordings, we identify more than 1,200 whistles. We identified specifically 9 categories of variant whistle and 17 types of signature whistle. So here there are just some examples of the contour. In addition, we found that in larger groups, there was a significantly higher probability of signature whistle being emitted than in smaller groups. And also we found that when trawlers were present, there was a significantly higher probability of signature whistle being emitted, but also if calves were present. So there is this effect of this relation. Then we found again that in larger groups, there was a significantly higher probability of this specific type of signature whistle, the number 9, being emitted than in smaller groups. And then we found that in groups with calves, there was a significantly higher emission rate of inflection points in the whistle. So what can we say about these results? Regarding the variability of variance whistle, the up contour, so the ascendant contour, was the most predominant, and this is actually in line with the predominance of this specific contour reported in other studies for the whistle production of dolphins. And this is also in line with the fact that this specific contour is the most present in the Mediterranean Sea. So basically our results support the fact that the up contour could constitute a predominant whistle type that individuals belonging to the same group share. And in addition, since some studies reported that the up contour was associated with social behaviors, our case could be the same. So maybe dolphins produce this when they are engaging in social interactions. For example, during foraging, they need to coordinate between them. Then we also found that in big groups there was a higher probability of signature whistle being emitted, and this is in line with the general higher rate of whistles in larger groups that we normally found in literature. And we also found that signature whistle emission was influenced, as I said before, by the relation between trawlers and calves presence. So this actually is in line with some things in the literature. First, with the general influence of group compositions and trawlers' presence on the acoustic production of whistle in bottlenose dolphins. And it's also in line with the mother's tendency to emit signature whistle more frequently when they are with the calves, instead when they are not. So basically, overall, our results support the known signature whistle value, functional value. So, for example, to coordinate the group, to favor the cohesion between mother and calf. So, in dynamic contexts such as the ones that we had here, so big groups with calves, trawlers, signature whistle play a crucial role. Then we found that this specific contour was highly emitted in big groups, and we explained this in two ways. First, this specific contour could belong to a mother. So in big groups, it's more probable that bigger distances between individuals, and so between mothers and calves, happen. And so it's important for the mother to emit more frequently her signature to facilitate the reunion and to maintain the contact with the calves. But at the same time, we can also explain these results, suggesting that signature whistle number 9 could be a group whistles. And so not an individual contour anymore, but a stereotype frequency contour shared by the group. And so this would be consistent with what is emerging from the literature. So there are very few studies suggesting this. And again, in this case, the group whistle would have the functions of favor, cohesion, and coordinations of individuals in big groups. Finally, the fact that in groups with calves, there were more inflection points, so changes in the slope of whistles, could be actually an example of madres, namely the child-directed communication. Because, for example, in 2020, there was this study who reported that mothers, when they are with calves, modify the acoustic structure of the signature whistles. And so this could be the same, again, to keep proximity with the calves. So, to conclude, first of all, we intend for the next steps to expand this data set, because this is only a data set of only 1 year of data collection. So we would like to include further field seasons of the past, because we have other, but also present, because we are currently collecting data in the Ligurian Sea, but also in the Adriatic Sea, and future data collections. And then we intend to develop an acoustic capture mark recapture method using exactly the signature whistle type. So this would be a very passive monitored geographic units. And we also intend to expand this method in other areas such as the Comoros Islands that actually constitutes a incredible hotspot of biodiversity for cetaceans. And so, yeah, thank you.
Thank you, thank you. Do we have any questions? No. Okay. No one? No. I do have some questions. The last part about the inflection points, I think that's very interesting. So from what I understood, what you suggest is that these inflection points are here to trigger your attention, right?
Yeah.
Do you have other examples in maybe non-mammal species or like field mammal species that do have this kind of attention-getting thing?
Actually, I don't know from the literature, but we can also think, for example, when we talk to children, I mean, we in general, maybe we alter our voice. You know, maybe we higher the pitch, and so maybe this can happen also to other species, especially mammals, since they share the same phonatory system. So, I mean, in the cetacean it's different, but yeah. Okay.
Good. I think there must be some things.
Otherwise,
I know in birds they are. So
yeah.
Should look at it. Good. Great. And just to be short, so the boats, are they supposed to be some kind of a threat to the calves, or do you think they are? Is it like a dangerous situation for the calf that would explain the increased, communication with the mothers?
But do you refer to the trawlers?
Yeah, sorry,
trawlers.
Yeah.
Actually, I don't know, but I mean, in this area, I mean, they monitor this population from 2001, and they consistently see that dolphins follow the trawlers, even though they have calves sometimes. So, I mean, if trawlers would be a threat, maybe the mothers
avoid it. Yeah.
So maybe the advantage of finding concentrated food of source is higher than the effects of noise and
All right. Do we have any other question? Yeah, just.
Hello, thank you for your presentation. It was really great. I was wondering if dolphins, and especially male dolphins, disperse. And if it's the case, if they alter their vocalizations to increase coordination in new groups or stuff like that.
Well, actually, there is some evidence that males dolphins can modify their signature whistle, for example, copying the signature whistle of other individuals, or when they form alliance between males. So yeah, I mean, this could happen, but we don't know because we cannot see it's difficult to identify who is emitting. So in fact, another goal we have is to try to match the signature whistle with the dorsal fin. So if, for example, every time we find same individual identified with the dorsal fin, we also find the recordings the same signature whistle type, maybe it can be that signature whistle type corresponds to that specific individual, but of course we need more data to assess this.
All right, thank you very much. Good. You can keep that. Yes. Up to the next talk about subtropical forest of Northeast India, super cool. Let's go.
Hello, good morning, guys. I know we all are feeling very sleepy. Even I am also feeling very sleepy. But yeah, I'll try to make you guys awake. Okay, so my talk is about passive acoustic monitoring on the Ensiferans, calling diversity in the northeastern part of the India. So I'll just first come to Ensiferans. I just want to know who all know about Ensiferans. What is Ensiferans? Oh, only 1-2 person? Oh, okay, cool. So Ensiferans are actually a group of orthopterans So the Orthoptera order is divided into two suborder, Ensiferan and Califera. So Califeras are all the locusts and grasshoppers we saw, and the Ensiferans are all the is our bush crickets. They comes under that. And Ensifera is again divided into two, Titigonidae and Grilloidae. So Titigonidae is a bush crickets and Grilloidae is the ground crickets. So yeah, there is good diversity there. So just some GK about them. So usually the male, they produce sound, and like by vibrating their wings, by vibrating their wings in the mostly the forewings, and they usually use it for mating and advertising. And another interesting fact, their songs are usually genetically encoded. So it's not like birds, like they're learning it. So that's very interesting. And the females are tuned with their own conspecific calls. So that's how they understand. And yeah, another thing is we all talk about when the passive acoustic monitoring for the ecosystem health and the forest health. Ensiferan can be a good indicator to understand the forest health. So that's why this project was we are thinking for. So little more about Ensiferan for the tropical forest. So when we talk about crickets here, everyone is like, something in the ground makes sound. Yes, in a ground makes sound, but it's not only in the ground. If you go to the bushes, they are in the bushes. If you go to the canopies, they are in the canopies, and you can see how beautiful they are. Like huge, big leaf-like structure, and then the another one which is a tree cricket in the top. So it's actually doing baffling. So they make a hole inside a in a leaf and then baffle it there. So can it go for longer distance? Yeah. So now will come more details. So how our Ensiferan calls look like? So usually they have like basically two types of calls. So one is like simple kind of chirping calls where they have syllables and pause, syllables and pause, like that, or a trill where continuous syllable without any pause. So, and some of them have combination of both. So that is the fine temporal structure. So if you come to the spectral structure, there is also varieties. So some of them are very pure tone calls. So they are like very low frequency. They here you can see it's a 5 kilohertz and then harmonics. And some of them are broadband. So they are like almost 2 kilohertz to 80 kilohertz. Some of them have a higher frequency, very high frequency. Their main call is at 40 kilohertz. So it's that much diverse. So yeah, so let's come to a very real picture of what's happening in a rainforest. So you can see there is two conspecific callers. They are calling, they are communicating with each other. But no, it's not only that. There are more. There are more. There are more. I think you need to play the sound from there. You need to click. It's not. Sorry. Yes. So it's just a few second recording, and you hear that many. And now you think there is a predator. So they are also looking for these calls. So can they catch the crickets? So it's a high competition zone. There is a high competition, intraspecific, and also the from the predator pressure. So it's highly diverse. So just to understand how diversity of one subtropical forest in India, which is very less explored in India. So those are my objectives, like see the acoustic community assemblage, and then see the acoustic niche partitioning between all the callers, and acoustic space used, and call occurrence and call type composition. So let's come to my field site. So this was Nongkhailam Wildlife Sanctuary. It's a subtropical rainforest which have both kind of trees like vegetation, tropical, evergreen, semi-evergreen, deciduous, everything there, and it's high humidity forest. So this is actually in the Meghalaya district, you can see. So that comes under the Indo-Burma biodiversity hotspot. And this was my location, these two transit sites I picked in the for recording. And this is a rain graph there, and based on the rain I divided into 3 seasons: pre-monsoon, post-monsoon, and dry season. Monsoon, it's so heavy rain, so I can't record anything, so there is no data from there. Okay, so what we did, so first of all, I don't have as many that many of audio mods, so I have like couple of audio mods, so I can't like leave them there. So it's like a passive acoustic monitoring, but little bit of active way. So like I have to do the transect, like every time I have to put here one, then when the recording is done, I have to move it to another, then I have to move it to the another. So that's how I did. So it's a 500 meter transect. So both the transect are like that, and there is 10 segments in it, and you can see this is the audio moth, and we recorded in 48 kilohertz sampling rate. That's mean it's can record up to 24 kilohertz. So beyond that, if there is any call, I don't know, sorry. So okay, it was recorded only in 8 p.m. to 12 a.m. And another thing, it's all data is manually annotated. So there was no AI. So we don't have the facilities in the universities with the computer science like labs, like we can do AI and all, and we don't have the database. So yeah, so these are the limitations. So unavailability of the Ensiferan call database in India that's why we can't go for AI. And the second thing is all recorded passively. So I don't know what species it is. So I give all the call a call ID, like call ID 1, call ID 2, like that. And yes, so I already talked about it. So you need to, I think, play from there. So this is how a spectrogram look like. There. There throughout the whole spectrogram, there is multiple callers. So that's how our spectrogram look like for us. So so we quickly we got 33 different callers, and if I come to the acoustic assemblage, so based on the frequency, we find out that the most of the callers are in the low frequency and low bandwidth, and some are in the high frequency. And if I see the seasonal variation, there is a seasonal variation based on syllable repetition rate, which is a fine temporal characteristics, and the frequency in the x-axis. Based on that, like dry season have very less diversity than the others, and you can see there is in the lower frequency there is lots of overlap. So most of the callers are belong from the lower frequency. So then we wanted to know how they are separating, like if they all of them are in our same frequencies, so how they are hearing each other. So we did a PCA for that. So we now categorize them like trailer call types are different, high frequency and low frequency are different. So trailer call types are nicely separated out from each other. So even they are in same frequency, but because of their fine temporal pattern, they are different. High frequency is also the same, but in case of lower frequency, there are still lots of overlap is there. So still we don't know like how it's happening there. So we need to do more deep work on it, how they're separating each other. So now if I come to the acoustic space, sorry, there was a season. So the first one is a dry season, then pre-monsoon and post-monsoon. So in acoustic space map, usually in this graph, what was like each beans is about one frequency, and then you can see the red, more darker color means the more highest frequency was used in this place. So we find out that there is a variation in the acoustic space used in the season. So now we come to the occurrence. So occurrence is talking about like how many times that call types are present in each transect. So, and is it difference in the season? So we find out in different season, they are actually varying. So it's like some callers are present in this season, but not in this season, like that. So we find out there is a variation in that, in the both the transect. And call type composition, where we saw like how many total call types are there, and in each call type, how many occurrences are there. Is there is any kind of variation with the season? But the p-value you can see is very low, so we didn't found any call occurrence variation in the season. So yeah, so these are the conclusion. And yes, obviously the passive acoustic monitoring is a very good way to understand the call diversity and the ecology of the Ensiferan calls. So in a future direction, I really wanted to work with develop more Ensiferan call database with collaboration, the AI work, if someone is really wanted to collaborate on that. And it can be a long-term monitoring project. So I wanted to thank my lab members and my PI, Dr. Bittu, and other all. And yeah, thank you.
Thank you very much. You have, yes. Oh, great.
Hi, very interesting talk. I have like two questions. One is, I probably have more. One is about like the sampling rate that you choose. Why did you choose such a low sampling rate? Was that a reason for that?
Yes, so as I already said, I have only one audio moth. So what is happening if I'm keeping an audio moth here, and if I give a very wide range of sampling rate, so then the lower frequencies callers who are usually in the ground, they already get masked by the higher frequency calls. So that is a problem, because we have like a variation of the frequency callers. So I wanted to like keep till one range, because if I want to cover all of them, then in a spectrogram what happen, if some recordings are in 250 kilohertz sampling rate. So what is happening, the low frequency callers are so faint, so they are not visible. Okay. So, and you can see in this graph, most of the cases we have more low frequency caller than high frequency callers. So I'm like, okay, let's stick to that. We can do it later, maybe only for the very high frequency callers.
And when you talk about like having more overlap in the low frequency range, have you don't have any data on the on where are they calling?
Yes,
part of the.
Yes, that is the one thing we're looking for, because we place the audio moth maybe in like 6 feet or 7 feet height, so we don't know that vertical stratification is there. Maybe they are vertically, they are all separated already. So that's the one thing we need to explore, like how it is happening. Yeah.
And at which temporal scale, sorry guys, like at which temporal scale we're looking at? Because when you see the spectrogram, they seem like very, I love it because they seem very organized, like in the frequency. But then you say like there is overlap in there. So like, are you looking into which temporal scale are you looking at? Because if you think like, okay, in one night, like in a night, or even like in a season, yeah, I have this many species calling, like overlapping the same frequency. That's one thing. But then if you look like at a short scale, they might not be overlapping.
Yes, that's also a right question. So yes, we are getting sometimes like in one season, in one night, they are overlapping with each other. So there is a spatial difference, like maybe this night they are having, maybe in this season, and the another season they are not. But some of them are literally like, especially the broadband one. So they are like literally one tiny dot in this broadband. So users already get masked, and sometimes they're like two together. But yeah, if you go with the duty cycles, so if you know what is duty cycle, so duty cycle wise, you can still make the difference, because what is happening, the lower frequency, they are having either a higher duty cycle or lower duty cycle. Like lower duty cycle like t, 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 like that in 4 kilohertz, and the higher duty cycle. So they both will be in the same, but one have higher duty cycle and lower. So in that case, we need more focal recording to say, ki, okay, the two are different.
All right, thank you very much. Perfect. And now we are going to Colombia. Great.
So hello everybody. My name is Diego. I'm part of the Universidad de Rosario, but I'm working on an independent project funded by ProAves Foundation, and it's called the Vocal Behavior of New World Migratory Warblers in Wintering Seasons. In wintering grounds, sorry. So let's start with what's bird migration. It refers to the millions of birds that fly from the northern hemisphere to the tropics during their winter. So these movements and the changes are linked to the bird's life history and ecology and behaviors. So in the tropics, they only need to survive and forage, while in the breeding grounds in the northern hemisphere, they need to do that, but in addition, they have to do breeding tasks like courtship, nest making, they lay the eggs. So there's more behaviors. Also, I want to highlight like Colombia. If you don't know where Colombia is, it's right here. It's right upside the equator. And it's a really crucial hotspot because it hosts more than 150 species of migratory birds. And because of their wide range of environments and their food availability, it's like really important. So now, what does that have to do with vocal communication? Since there are differences in the behaviors and ecology, one would expect that the vocal production changes as well, because there's different behaviors. For example, that's the main thing that people think is that in wintering grounds, birds don't vocalize that much because they don't have to do courtship, they don't have to establish breeding territories, so they reduce a lot or ceases the vocal production. So I want to see if that's really happening or it's just an assumption made from like the temperate zones. So I use this family, Parulidae, the New World Warblers, which is a songbird family that are Osin. That means that they learn their songs because not all songbirds learn their songs. Yeah, and it has 115 species, with almost half being migratory birds that winter in the tropics. Their vocal system is quite simple. Some have one category of songs or two, and some species present more complex songs as well as flight calls and alarm calls. So with all this in mind, I've had the objective to see what's the vocal behavior of this family in the wintering grounds and see these characteristics of the vocal behavior, which species are vocalizing, what types of vocalization, their acoustic parameters at the times, how frequently, in which habitat, and the ecological context. So a lot of the characteristics of the vocal behavior, like baseline for these birds. So I went to 2 different reserves for 1 month each to cover different migratory paths. This one covers the Central American migratory path, and this one covers the Caribbean migratory path. They have similar characteristics, and they're known to be hotspots for migratory species. I use both passive and active acoustic monitoring. For passive, I use Audimoth, and I recorded 6 hours daily, 3 hours at dawn and 3 hours at dusk. And I placed them in 3 consecutive days in the same transect and then changed the transect. And for active acoustic monitoring, I use a directional microphone, and I did opportunistic direct recordings to birds actively vocalizing. And I documented the bird characteristics like sex, age, where they're vocalizing at what times. Then for the analysis, I used BirdNet, obviously. Everyone knows BirdNet, but in South America we also have it, and we have over 2,000 species for detection. I use a 0.3 confidence threshold because this is in the tropics is really hard to do detection because there are a lot of environmental noise and pollution like crickets, cicadas, and running water, wind. And then with all the detections, I filtered the migratory warblers, and I manually reviewed each one to compare them with the vocalizations known from the temperate zones and see if they match. And then I measured the acoustic and contextual features of each one. So what did I find? For active acoustic monitoring, I only got 7 recordings, 3 from Canada warblers and 4 from morning warblers, more on that later. And for passive, I got 320 recording hours with over 36,000 matches in all the recordings over in 742 species, which you can see it's a lot of species. But then after filtering migratory warblers, I got 11 different warblers with 189 matches. But then when I manually checked it, I ended up with 5 confirmed species of warblers vocalizing, which I'm gonna present like shortly. So the first one is blackpoll warbler, Setophaga striata, which I got 6 detections, and all the vocalizations that I found was rapid trills. You heard that it's like a really high-pitched trill. They're described in the wintering grounds in male-male interactions to defend breeding territories. So they also in Colombia. Then the Canada warbler, I got 52 detections, and I found both calls and songs. Calls sound something like this. And the songs are. So I found it seems like the mode 2 songs, they have mode 1 and mode 2 songs. And these are described in pair males and fledglings and territorial interactions. And also call notes, I found it given by both male and females. And there are, the function is basically to alert mates or conspecifics about danger. Then the morning warbler, Heotrypis philadelphia, I got 85 detections, and it was the bird that I found more detections. I only found calls, sounds like this. And these are described to be given by both male and females when predators approach nest or territory, so another type of alarm call. Then another water thrush with 15 detections. I got also the chin call. And they're actually described in wintering grounds, and they're described to defend foraging territories as well as location to conspecifics. And finally, the Tennessee warbler, I had 5 detections. I found 2-part songs, which are these ones. So these are not really common and not being described too widely in breeding areas, and they're only given by male and may be related to defense of the territories. So the main question is, why are they vocalizing these species? Because they're not breeding, they're not defending breeding territories, they're not finding couples. So the first is the ecological standpoint, that they may establish and maintain and defend temporary territories to forage. So I have this territory that is mine, and that's where I'm gonna find my food. The second one is a physiological point, where birds before migration have a peak of testosterone, male birds, and they may have residual testosterone in the migration that may enlarge vocal control regions and result in singing. And finally, from a behavioral standpoint, birds may use these vocalizations to distinguish between neighbors and strangers in inter or inter-specific flocks. That's to say, I want to find the bird that I know, or my family, or my friends. Also, birds, males particularly, may practice and refine songs to improve them in preparation to the breeding season. So I want to practice my song, so when I'm going to go breeding, it's better, and it has it's better for females. And finally, fledglings may have their period of vocal learning. These are species that have vocal learning. So the adults may be teaching the fledglings how to sing. So to conclude, I think one of the most important things is first see how it's important to understand vocal behavior, to know about different things like life history or estimating population presence, and help with conservation efforts. But most importantly, I want to highlight the importance of Colombia and other tropical zones as important study sites, because if we only are studying birds and maybe some phenomena in temperate zones, we're only getting half of the story. So for us, you to know that it's important also to see what's happening in other parts of the world, because maybe the phenomena are not explained the same. And thank you everyone for listening. I want to thank all these amazing people that helped me in all of the steps of the presentation. Also, I'm searching for a postgraduate, so if you know something, you can reach me. You can write me my email, or you can like come for me and talk to me. So thank you, everyone.
Thank you very much. Do we have any questions?
So it can be a stupid question, so I'm sorry, but I worked on data on tropical sites, and I learned that there could be flocks of birds. Is it the case when they are wintering them and there? And if so, can it be some kind of cohesion of the flock, calls or something like that?
What I found, I didn't really find many like migratory species vocalizing at the same time with other migratory. I've known of some flocks, but I only think they're from the migration, not the like they're staying and foraging. I think they're separate and established territories. So normally they are not flocking together like during the wintering like periods.
Thank you very much.
Yes, thank you. I was wondering, were you expecting to find more than 5 warbler species? And if so, what do you think kept you from detecting them?
I think not. I was actually surprised because if you like look at Senocanto or the Macaulay Library, you can find a lot of vocalizations of migratory species in tropical zones. So I was really surprised from some of them. Maybe the places are not the best because there are some birds that I'm gonna go back to the map. There are some birds that don't fly all the way through the tropical zone. There are some that stay in the Caribbean or something that stays in Central American. So maybe there's some birds that there's not all of the migratory warblers wintering in Colombia. Some of them go even lower. So maybe if you change the spot of the location of the recordings, you will find different things and different species.
Do I have the time for one question? I do have one question. So if I understood correctly, you first put it into BirdNet and then you checked whether it made sense, right? Do you think that, because I guess BirdNet does not know everything about this repertoire, because that's what you're trying to understand, right? So could you think that maybe BirdNet missed some of the variation in the calls and maybe, detected only the songs and not the alarm calls or things like that, and that could have been a bias for you under detecting some species?
Yeah, definitely. I've thought about it a lot because if they're singing something different that the vocalizations that we know in temperate zones, so BirdNet wouldn't be detecting it, because mostly BirdNet is trained on these species, in the migratory species, in the temperate zones, because that's the bulk of information that we have. So maybe I have some of the calls or the songs not detected in the wintering. Yeah.
Okay, thank you. So no, I think we're good. Good. Thank you very much.
Thank you.
C'est bon là? Okay. So, hello everybody. So we'll now have a session about acoustic monitoring, and we'll start with Fanny. We'll talk about bat flight. The floor is yours.
Hi, I'm Fanny Aguilera, and today I'm going to present you the results of my master thesis, supervised by Benoît Charras and Kevin Barré. Basically, I'll try to answer to one question: how many bats is too many? Through a 3D flight path study on density and hunting success. So the optimal foraging theory suggests that animals maximize the balance between net energy intake and foraging costs, which can be hard in such an unpredictable environment. And that's why sociality can represent a strong driver of foraging success and an interesting strategy. Because as individual density in a group increases, the foraging success can increase thanks to facilitation, thanks to information transfer, for example. But after one threshold, competition can happen and foraging success can decrease because of acoustic interferences, for example. And that's why we can talk about a social foraging trade-off. Such trade-off has been mainly studied in easy to monitor and journal species. However, bats, even if they are nocturnal animals, are interested to study for such interactions because they have a variety of social structures, a variety of foraging strategies, and they are globally distributed. But also they use a lot echolocation to find prey and to flight, which is an information vector, which is really interesting for bats to share informations. It's also a frequent behavior, easy to monitor behavior for scientists, and it's really interesting because inside the sonogram we can see all the different behavior of bats while hunting. Because echolocation is a sensory system for spatial orientation and prey detection. So we have a first phase called the searching phase and characterized by those searching calls, followed by a feeding phase characterized by this precise pattern, the feeding buzz, which are followed by a post-buzz pause before the start of the next searching phase. And those post-buzz pause are really cool because they could and they are an acoustic indicator of predation success, with as prey is being captured, post-buzz pause increasing because of the handling time of the prey and the showing time. So that's really cool. And that's why I want to answer that question. And I answered that question for my master thesis. How does bat density affect predation efficiency, considering conspecific and heterospecific? And for predation efficiency, I considered the capture attempt, so the foraging effort, thanks to buzz, and the capture success, so the foraging outcome, thanks to the post-buzz pause. Also, the predation efficiency can be modulated thanks to the foraging strategies used by bats, being a decrease in flight speed as they attempt to capture, and an increase in sinuosity to increase maneuverability and potentially the capture success. And that's why I also decided to take the flight speed of the bats in my models. What did I do? So I used a microphone array to record echolocation calls of bats. That microphone array is composed of 4 different antennas, and the calls of bats arrive at different times in the antenna, allowing us to calculate and to obtain the XYZ coordinates of the emitter. And then, thanks to different criteria, I can link the points together and obtain a trajectory for which I calculate the minimal speed of the bat and the co-occurrences. So for example, here you have different trajectories, and you can see that for this one, you have 2 different co-occurrences, so 2 different individuals being present at the same time. Then I use the echolocation call to identify the species, thanks to Tadarida, and to obtain a buzz score obtention, so sort of probability of presence of buzz in the audio sequence, thanks to Tadarida C. I selected high scores, so high probability of presence of buzz, to recheck around 2000 audio and measure the post-buzz pause duration to have this proxy of the capture success. So to resume my variables, I have the co-occurrences for conspecific and heterospecific for each trajectories, also the minimal flight speed of the bats, which is kind of a proxy of the foraging strategy, the buzz score, which is a proxy of the capture attempt, and the post-buzz pause duration, which is a proxy of the capture success. And I'm only going to present the result for the median range echolocators, so essentially Pipistrellus, because they were highly represented in my datasets. First of all, I tried to model the effect of conspecific number on the buzz score, also with the minimum speed as a proxy of the foraging strategy, like I said. And I obtain a negative significant effect of the minimum trajectory on the buzz score, which is quite logical and predictable concerning our hypothesis because they increase their maneuverability thanks to that foraging strategy. Then, when modeling the number of conspecific, I also find a significant effect with first a positive effect and after 7 individuals, a negative effect of the number of conspecific on the normalized buzz score, meaning that they tend to buzz at up to 7 individuals being present, and then they decrease their probability of buzz, which is also quite logical concerning our hypothesis. We can observe a sort of trade-off with first a facilitation effect thanks to information transfer, eavesdropping, which is really present in bats, and then a competition effect because of acoustic interference, or jamming, which are active acoustic interference, or limiting prey on site. And then when looking at the interaction of the number of conspecific and of the speed, we can observe that at high conspecific density, the slowest speed, so the green ones there, are the one that keeps a high buzz score. That could make us think that bats, they maintain their foraging strategy of decreasing the speed and increasing the buzz score at high conspecific density to fight against this high conspecific density and increase their chance of capture. Then I modeled exactly the same, but for heterospecific, and I found the same results. So a negative effect of the speed and a quadratic effect of the heterospecific number on buzz score. But why do heterospecific has the same effect than conspecific? It's really interesting, and one of the hypotheses could be the foraging similarity hypothesis that says that two individuals that chase the same prey find valuable information in the calls of the other individual, meaning that if Pipistrellus emits a buzz because she finds a diptera, it's worth it for Myotis to listen for her and to know that the prey is present and then to come at the foraging site. And that's why we can see the same effects for heterospecific sharing the same prey. But no combined effect of both, no interactions, suggesting that there is no adaptation with heterospecific and at high heterospecific density of the foraging strategy. Then I modeled the post-buzz post-duration, so this time is the proxy of capture success on the effect of conspecific number, on the capture success, without the speed, thanks to a model selection. And I found a negative effect, a significant negative effect of the conspecific number on the capture success, which is quite cool. It's a kind of competition between conspecific, and when modeling the same with heterospecific, I did not find the same results. So why this time do we have divergent effect between conspecific and heterospecific comparing to just before? Potentially because this is a real measure of competition between individuals of the foraging outcome and not only of the foraging attempts. So competition can be more precise with those kind of metrics. And as conspecific share similar prey, more similar prey than heterospecific, they also tend to have more acoustic interferences together and tend to increase their vigilance as they leave the roost together, for example. It could lead to an increase of competition between them. So to resume everything, I did find a trade-off when looking at the capture attempt together with the flight speed of the bat density on bats. But I did not find any trade-off and only a competition effect when looking at the capture success and only with conspecific, showing that both metrics are really complementary to measure how many bats is too many. Actually, we could say that 7 bats are too many when looking at the capture attempt, but it's not the case when looking at the capture success, and that's why such metrics are cool. Some methodological perspectives I should have, and it would be valuable to distinguish the feeding buzz presence and absence, and not only looking at the scores. For example, by doing a regression like that, I could set 0.4 as a threshold to consider buzz to be present or not, and it could modulate my results and give other insights. I could also classify the capture in success or in fail according to the post-buzz post-duration and not only looking at the distribution. Because when you look at the distribution of this post-buzz post, you can observe 2 different peaks. The first one could be characteristic of a low success and the second one of a high success. And it's quite logical when looking at the references. For example, from Myotis d'Antony, the literature says that 100 milliseconds could be the threshold to consider a capture to be successful or not. So I could have classified those attempts, like I said, but it's still hard to do because there is a lot of heterogeneity in those values, so it's hard to do. And finally, and my main goal would be to automatize the post-buzz post measure because it's quite long to do by hands and maybe by machine learning. It could be an option. I don't know. I'm not good with that. But ideally, I should be able, and the machine learning, I hope, would be able to find a buzz and measure this post-buzz post, but it's quite hard to do because, as you can see, there is a sort of shift in the frequency after the buzz, and before the next echolocation call. So it should be able to see such patterns and also to deal with overlaps. So I don't know if it's possible or not. You can come to me later if you want to talk about that. And finally, and some perspectives about my work to come in a PhD. So to model even to get insight even more on this foraging trade-off, other parameters need to be taken into account, such as the prey density, which can be really limiting, as we said. So we're going to measure prey density on site while doing those measures. Also, the habitat can modulate a lot species advantage and benefits on site. For example, pipistrellus can be more at ease in open habitat compared to myotis. And finally, light pollution, because it's a big problem and pressure in our modern world, should be integrated in our models, especially because you can also modulate the benefits of some bats compared to others. Thank you for your attention.
Thank you. Any questions?
Thank you, that's super cool actually. I was wondering if you could have a bias, because if you have more individual, perhaps it's more difficult to detect post-pause pause, because then you have more noise in general.
There is definitely a bias when we have a lot of individual in our audio sequence. It's way harder to measure this post-pause effectively. But also we want to search for high density places to obtain this gradient of individual and look at the trade-off. And potentially that's why we didn't find any quadratic effect, for example, when looking at the capture success, because we didn't have a beautiful gradient of individual. But yeah, it's a choice to do, whether take not dense place or really dense place and have this trade-off potential to measure.
Very nice talk, thank you. That was super interesting. I think you began to answer this question, but for now, the sites you studied, they were all similar, or what kind of sites was it?
So I'm not the one who did the recordings. It was Kevin Barret, which is my supervisor, and he did try to normalize the site where he did the measures with the same distance at the first aquatic place, the distance at the first I don't know how to say it in English. What? Edge. The distance to urbanization and stuff like that.
Super cool.
Yeah.
Yeah, thank you. I was wondering, I'm not sure I understood what you mean by interferences. Like, do you refer to the physical wave theory phenomena where there is additive and destructive waves? Is it this, or is it more like another concept but using the same word?
I don't know if I can come into the physics of the interferences definition, but it's the fact that when individuals vocalize together, they can struggle to hear their own vocalization coming back to them, and it's the way echolocation works. They wait for the call to come back after the echo. And as there are plenty of individuals on site, it can be more difficult to find your own.
Okay, because like interferences is quite well defined in wave theory, and it's not exactly that.
Okay.
So maybe there's like a term, concept maybe to define around that.
I should search more into the real definition of interferences now.
Thank you.
Okay, so thank you very much.
Thank you.
Okay. Now we'll have Alexandre. Okay.
So, hello everyone, I'm Alexandre Hammoudi, a master student, and I'm going to present you the work I did with Léa Mariton, Stéphanie Bourgeois, and Marie Sigaud on the contribution of eco-acoustics to the protection of a critically endangered rainforest species, the African forest elephant. It doesn't work. Yeah? How does it work? Okay. No. And so, as we know, human activities can have an impact on biodiversity, whether through overexploitation of resources or destruction of habitat, like deforestation. And in response to these impacts, a lot of conservation biology studies are being carried out. In parallel, bioacoustics has been and is still a fast-growing discipline. So bioacoustics revealed itself as a good, valuable tool to use in conservation. And especially because it's a non-invasive tool and it allows long monitoring and study and allow to study some species that are difficult to access. And so during the 2010s, there was a lot of large data sets. So bioacoustics has been more and more coupled with CNNs. And it's in this overall context that our study species find itself. So the African forest elephants, it's a species that live in group in the rainforest of Central Africa, but it also frequents some open space like clearings and salt pens. So salt pens are clearings where elephants can find minerals. And as you can see on this map, the populations has greatly reduced during the last decades. And so now 60% of the remaining populations live only in Gabon. So Gabon is a Central African country which is covered by 88% of forest, but 60% of these forests are subject to logging permits. There is also poaching pressure in the whole country and conflicts with humans because elephants eat crops. So in response to these impacts, the government and the National Park Agency are setting up a national action plan to the protection of elephants. So all these prompted us to ask the following question: how can bioacoustics help us understand the impact of human activities on the presence and behavior of African forest elephants? And thus we used the potential and the performance of neural networks to detect vocalizations and the poaching pressure in our datasets in order to test our hypothesis. The first one concerned the impact of human activities and the seasonality on the intensity of area used by elephants. And the second one concerning the impact of human activities and seasonality on the daily activity patterns. So an acoustic monitoring study was conducted in the southwest of Gabon. So it's in an area where there's high density of elephant, and it's within and around the national park of Mukalabadudu. So we had 4 types of sites. The first one was the national park. In theory, there was no human in the national park. There were the clearings, or salines in French, and they are at the north of the map. There are the concession with the selective activities exploitation within, and the village with the constant human presence. For each site, there were 2 recorders that were placed at 2 kilometers apart for the list, and they record the 10 first minutes of each hour with a sampling rate of 8 or 16 kilohertz, depending of the recorder. So for the vocalizations that we detected, there were raw trumpets and combination call, which are the combination of the vocalizations that you can see. They are mainly used in social context, but there are also lower vocalizations like the rumble, which are the main vocalizations of elephant. They used it a lot. And there are the rumble low frequency, which the human ear can't detect. So to assess the poaching pressure in our dataset, we used the Kogu neural network, which was specifically trained to recognize gunshots in a similar area as ours in Gabon, but it didn't perform that well. So we used around the 300 gunshots that we have in part to train another dataset. We also used the false positive, and within this false positive, we had vocalizations. So we trained a model on BirdNet, because the automatic classifier on BirdNet allow us to training on on vocalizations of elephant and also on gunshots. And after 2 training, we obtained a final model. But in parallel, we also used Grumble, which is a CNN developed by the Cornell University as part of the Elephant Listening Project, and which is specific to the recognition of rumble and low-frequency rumble. So to test the performance of our model, we used a test data set, which consists of a random selection of 16 hours annotated per station. And the aim was then to use a binomial regression, which allows us to associate for each detection, its validation, and its confidence score. And then to obtain a detection threshold at 50%, which means that for each detection, they have at least 50% chance to be valid. So for the statistical analysis, for the intensity of use, we used a generalized linear model with negative binomial distribution. In factor, we used the site and season with an interaction between them. And for the daily activity patterns, we used a comparison of density curves analysis and between the gunshots and the vocalizations on each site. So for the results, we can first of all say that thanks to this protocol, we obtained 4,900 hours of recording. For the detection threshold at 50%, we only obtained 1 with Burnett for the trumpet in clearings. For the other site, it clearly underperformed, and especially for other vocalizations like Rumble. So Grumble was very useful because thanks to Grumble, we obtained a detection threshold on 5 of our 8 stations. It didn't really work on concession that you can see on top, but for the other, it was good. And so thanks to this data, we could perform our model. So for the first model, we had overall 9,296 total rumble, which equivalent to 13 rumble per day. We had significant results for the site, the season, and the interaction between them. And overall, we had significant results that we observed thanks to pairwise test, except for the seasonality on 2 of our stations, which was not significant. For the daily activity patterns, we obtained significant difference in the distribution of the gunshot curve and the vocalization curve, and it was even more significant in the clearings, so it's the 2 at the top. And you can see in blue the peak of activity of elephant, which are more on the nocturne, and in the night. And overall, we had difference in the distribution for the 4 case. So regarding to our performance of the performance of neural networks, we can say that we had too many high confidence false positive, which prevent us from having a lot of detection threshold. And so it questioned the generalization of neural networks, especially when we see with the Kogu, which was trained on similar dataset as ours, and it didn't work well. And in perspective, especially for Burnett, we have the idea to accelerate our low frequency sound, which could enhance the detectability, especially for the rumble. So for the prediction and the first hypothesis, the first prediction wasn't supported because in fact we obtained more activity in the clearings, despite having a high poaching pressure. But then it shows that despite there is a poaching pressure, the clearings remain attractive for the elephant because it's a social place for them. And for the second prediction, it was supported. We obtained more activity in the clearings during the rainy seasons. This is what we expected, especially when we know that elephants follow where there are fruit trees, and they are mainly there during the rainy seasons compared to the dry season. And for the daily activity patterns, we observed nocturnality in the clearings, which explain why they are still there. In fact, they just avoid the hours where the poaching pressure is at the peak. And we also don't see this behavioral adjustment in other sites, only in the clearings. In the park and in the village, we don't observe that, despite having a poaching pressure in these sites. And finally, the use of trumpets is interesting because this is vocalization mainly used by male elephants, and could suggest that the behavioral adjustment is even stronger for them because they are the main target of poachers. And to finish, we can say that the use of bioacoustics and neural networks help us to obtain important data for species that has very high conservation concern. Still, we want to improve the performance of our neural networks, because for now it's not that good. But this data will be used in future study and will support the anti-poaching efforts. And to go further, we should couple these acoustic data with visual tracking. Thanks for your
Thank you. Do you have any questions?
Really good presentation. It was so interesting to see that you were able to modify the neural network to do what you wanted it to do. I just wanted to ask a question about your recording devices. You said that they were 2 kilometers apart.
for the list, yeah.
Yeah, I was interested to know how far the elephant grumbles are propagating, and was this a sufficient amount of recording devices?
So for the gunshots, there are no clear study, but we think that it's enough 2 kilometers apart to that the sound will not be recorded by the 2 recorders. And for the rumble, no, it's okay because they go like in the forest is really dense, so they go like 500 meters, maybe 1 kilometer is the maximum.
Okay. Yeah, that's interesting. Thank you.
Yeah.
Any other questions? Okay, I do have one. Regarding your gunshot classifier, like gunshot looks very typical sounds. So I wonder what other sounds in the forest it could be confused.
With branch with the branch when they crack. What we have sometimes water sound. Cool, yeah. I don't have many. Yeah, and some vocalizations of monkeys. Yeah, sometimes they're really low frequency of vocalizations could trigger the neural networks, and it will detect this as a gunshot.
Okay.
But I mean, is there not any differences with gunshots? For example, I don't know, just the sound pressure level or any kind, just to.
There's in the time there was, because the gunshot is really short, but the neural networks in the first place for the Kogu was really bad for the difference between these vocalizations, which last sometimes 3 seconds, which is more than a gunshot, and yeah, it was bad for this.
And why did you originally thought about BirdNet and not like I don't know, but developing your own algorithm for that?
Because first of all, we thought that BirdNet would be sufficient to perform, especially for trumpet and wah, for the high frequency vocalizations. But yeah, for the gunshots we weren't sure, but it's also because we didn't want to code we didn't have really the time to code their own neural network, and also because we really thought that the Kogu network would work, and it didn't.
Oh yes, okay, thank you.
Okay, just to add something, like BirdNet is also trained with environmental noise, like fireworks or things like that, so it should work well with. It's a good idea to use it for gunshot.
Yeah, also. Yeah, and we used a lot of false positives like storm extra, but yeah.
Hello, thank you. That was so interesting. Sorry if I missed this, but did you like manually validate every detection then? So every elephant call and every gunshot. I'm wondering how you got the like precision and sensitivity metrics.
Okay, so for this we annotated 16 hours for each site. Okay. So yeah, it was manually for that, and then I coupled the detection from the CNN with our annotation.
Nice.
Last question.
That's not really a question, but Alexandre also tried. We didn't know that at first, but in BirdNet, when you are trying to put a species list, you can choose gunshots. So actually, it is kind of trained for gunshots, but the results were even worse than everything we tried before. So yeah, botnet was not that good for that.
Okay, thanks. Thanks. And now we'll have Leah talking about insects.
All right. So yeah, oh, nice. I'm Leah Mariton. I'm a postdoc in the French National Museum of Natural History. And today I'll talk about the sound of wings beating, and to be more precise and maybe less poetic, on the combined use of eco-acoustics and deep learning to monitor pollinators in agricultural environments. So to begin with, it is estimated that about 90% of the flowering plants are animal pollinated. And this is really important for humans, as 35% of the world's food crop production comes from crops that depend on pollinators. And as many of you may already know, we do observe a declining trend in pollinators' population. This is because of habitat fragmentation, losses, chemicals such as pesticides, pathogens, exotic species, global warming, sometimes the management of domestic pollinators. And it's interesting to see that in every papers warning us about this decline, you can see sentences like those one. In fact, the authors also stress the need for more data, for more studies on pollinators, their decline, and the ecosystem services they are providing for humans through pollination. And the question is, how are we going to do that? So from now on, we'll focus on a taxa that is really important when you are talking about pollination, that is to say insects. And so to answer this question, you can take a look at this paper that is really interesting, that was released in 2022 by Van Klink et al. And they presented 4 emerging technologies that they thought would revolutionize insects monitoring. And among them, surprise, was acoustic monitoring. And indeed, for the past years, there have been more and more papers documenting how you could monitor the sound insects are producing to monitor them. And especially for Orthoptera, for cicadas. But you're gonna tell me, okay, that's quite nice, but you're studying pollinators and they're not proactively producing sound. So are you sure that acoustic monitoring is that a good idea? And likely, yeah. I mean, you all have this example of a mosquito buzzing in your bedroom while you're trying to sleep. What you're hearing is just the sound of its wings beating. And for bees, you can hear that too. Indeed, in 2022, Rodriguez et al. Showed that, 2024, sorry, that wing beat sounds of bees are related to species and individual traits. So knowing that, you could be eager to put other mosses all around France to monitor pollinators' activity. However, there are still many challenges to overcome. First, how are you gonna make correspondence between the buzzes you are recording and the species? Currently, we don't know how to do that, apart from the Balearic Islands. And more generally, how are you gonna detect those buzzes? So, as many of you here, our solution was to say, we're gonna use deep learning. Why not? And I mean, 2 or 3 authors already tried to do that with machine learning methods, such as random forest or supported vector machines. And the results were quite promising. However, as you may know, those methods are becoming a little bit obsolete because of the rise of deep learning. And you can say, no, now I want to try deep learning and convolutional neural networks, for instance. However, often it requires a lot of data, and we don't have those data yet. So this is what I'm presenting today. It's how I tried to construct a tool to detect those buzzes, and so to monitor insect pollinators in agricultural landscapes through acoustic monitoring. To do so, I had the chance to have access to data recorded with audiomorphs in the west of France, in the zone Atelier Plaine et Val de Sèvres.
In 2020, on 43 sunflower fields, and in 2021, in 28 rapeseed fields, for a total of more than 30 hours of recordings, so definitely not something you want to analyze manually. At least I didn't want to. So I had the chance, because before me, people already worked on this dataset. First, Ludovic Rochard already tried to do some annotation on the sunflower fields data. You have to know that in sunflower fields, most pollinators are bees, honeybees. So he annotated honeybee sounds, or the sounds. He constructed the first dataset that was used to train a tool that is called Tadari Deep, that is a new version of Tadarida. He ended up with a much more consequent dataset that was then used by Pablo Bolaños-Sittler. He deployed a new CNN, this time on both crop types, and he ended up with a final dataset with more than 6,000 sounds of pollinators, this time of a variety of species, but we don't know which one, and other noises. And this is the dataset I was given to work on, but I only had the image of the spectrograms. And I didn't want it to use Tadarida Deep. I wanted to try to use another application, and surprise, Burnets. So, as it has been told those past days, you can fine-tune the last layers to make it learn to recognize other taxa than the one it was originally trained on. This is what I really wanted to try to do. So to do so, I think most of you also had that. I had to do a lot of pretreatment of my data, a lot of cleaning that was really time consuming. First, I had to find the sound that were corresponding to my spectrograms. I wanted to try two things. I kept the data set as it were, and I tried to accelerate it to make the sound higher, let's say that. So I had two annotated datasets. On both, I had to randomly choose a segment of 3 seconds around my annotated sounds, because burnets require 3-second segments. I only kept one sound when several were overlapping. And so I ended up with my 2 annotated datasets having more than 1,000 buzzes. This was quite good, but we estimated that among them there were something like 75% of honeybees, which is great. We are also interested in this species, but this can be a bias. I divided my training datasets in my annotated datasets in a training one with about 80% of the data to train burnets, and I kept 20% of data that were not in the training ones and sites that were not in the training one to evaluate both my CNN, the one with the data as they were and the one trained with the accelerated data. So, what are the results? For the normal data, I ended up with this graph. So, as many also of you may already know, the outputs of CNN, like this one, is just detection associated to a confidence score. And you can draw the confidence score according to what the sounds really were. And in these datasets, you can see that in red, orange, yellow, those were the insect buzzes, just divided according to their fundamental frequency. So it was performing quite good because they had high confidence score compared to other sounds like car, dogs. However, there were still some issues, planes, motorbikes. And then I did so for the accelerated, the CNN trained with the accelerated datasets, and the results were much better. I did not have anymore the confusing detections with lorry motorbikes. That was really interesting to see. And another representation that we've seen for these past days is to draw the confidence, what the sound really is, insects or not insects, according to the confidence score. Then you do a binomial regression. This is the blue curve. And this binomial regression allows you to find a threshold under which you will discard the sounds or you will consider that they are positive detections. according to a constraint they can fix. For example, you don't want your false positive tolerance rate to be above 0.2. Once you've done that, you can calculate metrics. For instance, precision, the number of true positives compared to all the positive detections. Or the recall, the number of true positives compared to the total number of positives in your testing dataset. I did so for both my CNNs. I did the F1 score, which is kind of a synthesis metric between recall and precision. And you can see that the accelerated dataset trained the CNN was performing really better and quite good. So for the next step, I really want to deploy the CNN on my whole dataset, which is really time consuming. Yeah. And once we will have the detections, we could do the same thing, check a random subset and fix the threshold that we're gonna use, evaluate the efficiency, and if it's nuts, use it to test abundance hypothesis. We also want to work on embeddings. So for those of you who don't really know what it is kind of the numerical representation of your sound before classification. We want to try to project them, do some clustering, and we hope that some group will appear and we can try to work on a kind of functional acoustic diversity. So we are really waiting for that. Thank you for your attention.
Thank you. Any questions?
Thank you very much. That was very, very interesting. Quick question. How do you explain that by enhancing the pitch, it will get better results? Maybe it's...
So I have two hypotheses. The first is quite a simple one. It's that Burnett is trained on sounds that are not that low. So it might be better for sounds that are in the same frequency as birds. The other is that where Burnett, this is really a shame, but right now you can choose the parameters of the spectrograms. So by doing so, we change the parameter, and I think the frequency definition is better, and so it's performing better. But
okay, thank you.
Hello, I was wondering if there's some like rule of thumb you've come across when you're fine-tuning BirdNet for how many examples of the sound you need. Because I see you have like a lot of examples and you've come up with a good classification for the honeybees, for example. But yeah, as someone that might be thinking about training BirdNet, I have no idea whether I could do it with 100 sounds or I need 1,000 or 10,000.
So I'm still trying a lot of things before I find something that I found efficient enough. Before I was working on birds in the Amazonian forest, and I tried also a lot of things. Right now, I would say for kind of a binomial classification, having a little bit more noise is quite good. And I don't know, maybe the number of sound you would use would really depend on how characteristic your sound is. For instance, for pollinators, we had a lot of issue with motorbikes and voices, and maybe with 100, it wouldn't have been enough to have sufficient performance.
Thanks.
I was just wondering, could the problem, especially for the motorbikes detection and things like that, could it be solved just more by like looking at the recorder itself? If you consider, for example, the range of detection of your recorder, if you aim for getting a recorder with a lower range, since the buzzing might be more close than the motorbikes, definitely. Could this be a solution? Like a little bit like with dynamic range microphone. Or if I move a little bit, like it won't be recorded. See what I mean?
You mean, for instance, because the sound are not that, the amplitude is moving more?
No, it's not.
No, sorry.
Since it won't be in the range of the microphone itself, since it's far, there might be, I'm
not sure.
It won't be a problem anymore, you see what I mean? Because it won't be detected in at all.
I'm not sure it would work because we don't know yet, and it's something that if you're interested, please do that. We don't know the range of detection of pollinators. So a motorbike could be really far and have the same amplitude as an insect that is like 30 centimeters from the microphone. And from my experience, because I did also some annotations, I had really sounds I couldn't say by listening and by looking at the spectrograms if they were pollinators or motorbikes. So I'm not sure, but should give it a try, maybe.
Sorry. We'll have to move to the next speaker. So now we'll have Fanny, like this, Pauline, talking about nocturnal migration calls.
Okay, thank you. Hello, everybody. I'm gonna try to be quick because I think we all want to go have some lunch. But today I wanted to talk about a project, a citizen science project I'm working on with the National Museum of Natural History, like my two colleagues before me. I'm a bioacoustics engineer and I'm working on the project called Vol de Nuit, which is using bioacoustics and citizen science to monitor nocturnal bird migration. So, first of all, why do we use bioacoustics? Well, because we don't really have a choice. If we want to obtain species-specific data on nocturnal bird migration, other methods do not allow this level of precision. Also, during nocturnal flight, most birds that migrate above France produce specific calls called nocturnal flight calls. And 50 to 75% of bird movements in France happen at night. So this is why we wanted to study nocturnal bird migration. I think this is what we're all here for. So I put a few sounds, a few examples of the most detected species in nocturnal bird migration in France. First, you have the song thrush. Oh, okay. Yes. Then we have the grey heron, very different call. And then maybe cover your ears for the next one because it's the common moorhen. Yeah. Thank you. That's enough, I think. Yeah, so the nocturnal bird migration was already being studied by a few people here and there in France, but in 2024, the Ligue pour la protection des oiseaux, so this is the French BirdLife program, in collaboration with the CESCO lab, Biophonia and Trectelen, started a citizen science program to try and get all the data together. And how it works is we have volunteers all across France who record whole nights in their garden with their own recording equipments. It's continuous recording from civil dusk to civil dawn. They do a manual analysis of every single night, so it's really time consuming. They count the calls and identify the species, and then to try and take it into account the noise between sites, they put a recording usability. So, for example, if during 1 hour of the night there was a really strong wind and the night is 10 hours, then the recording usability for that night will be 90%. Here is an example of what the sound may look like. If you're lucky, you have birds, but most nights you don't have any birds. And this is what the data looks like when it's entered on the Trectelen platform. So it's a species list with the number of calls and exploitability. The network today, so it's a 1.5 years old, so it's still quite a young program, but we have a dynamic network of volunteers. This is all the sites that are active in France in 2024 with 106 volunteers and 112 sites. The coordination team gave 4 courses on species identification to the volunteers that wanted them. here are the groups and species that we focused on. And then 3 flash surveys were targeting groups or species. We're using a Discord server to communicate with the community. We have currently 250 members, and it's mostly used by volunteers to help each other identify the tough nocturnal flight calls. And then this year in 2025, we started providing recording equipment to new volunteers to try and expand the program. And we have the first few results. We had in 2024 95,000 hours of recording, 60 new sites on TrackTLN, and over 1 million flight calls that were recorded and identified of 151 species. Here you can see what's really interesting for us is here, no vol de nuit, so we're really happy. And here are the number of nights that were recorded in 2024 and the species diversity that was recorded by night. So obviously the efforts are mainly focused around the migration periods, so in spring and autumn, but they are recording all year round. And here are a few results of the flash surveys. So the flash survey on the Turdus groups allowed to detect 5 times as much song thrush as diurnal visual counts. And the flash survey on the ortolan bunting allowed one of our volunteer, Ryan Bosworthick, to find a new type of call that was, to our knowledge, never detected at night before. So this is the one. Yeah, you're just gonna have to guess. It's kind of the bloop, yeah. And here is a hourly rate of flight calls and a hourly rate of individuals that were detected by the program. All the plots here are by my colleague Paul Coiffard, by the way. And yeah, a lot of data, interesting. What's next? I'm finishing my contract on the project on Monday, but I will be coming back for a PhD project at in autumn. And I will be working still on vol de nuit using bioacoustics, citizen science, and AI to study light pollution effects on nocturnal bird migration. First, I will be working on the factors influencing nocturnal flight call detectability. So all of the variables that intervene between when the bird emits the call and when we detect it. Then I will be trying to implement either BirdNet, Chilpity, or a new trained version of them for the citizen science program to try and help them work quicker, because it's taking them a lot of time currently. And then I will be studying the effect of light pollution reduction measures on the nocturnal bird migration. Yeah, thank you.
Thank you. Any questions?
Thank you. Is it working? Yeah. Thank you for talk. It was very, I always find like very interesting to see citizen science projects and like how people were working for free a lot. But I'm just wondering how do they upload this data? Are they only saying like are they only saying this or this species was detected, or do you actually have like all the detections like marked on the sonogram? How's the?
Yeah, so for now, we're just working with the list of species that I showed you. So we don't have access to the spectrogram because we don't have the resources to store them, store the whole acoustic data of everyone. But we're working on a project trying to get a platform to get the spectrograms and the sounds.
Because I think even like Arbimon could work for that, not like because you can like everyone could just like draw like where the call is and just identify it. So maybe even because this is like a very rich data set if you have all the annotations, because we don't have that much data on the nocturnal calls, I imagine. I don't know. I'm just having some ideas.
I don't know about Albinon, but I'm getting it's a part of the project that I didn't talk about here, but I'm getting some of the recordings from a few volunteers to annotate myself and check the annotations of the volunteers against my annotations and against AI models such as BirdNet, Chirpity, and Perch. So it's kind of in between what you're saying, and yeah.
How did you associate the new call you recorded with the species since it's night?
The Ortolan bunting one?
Yes.
Yes. So our volunteer that discovered this call, he heard it in a time where there was a lot of other calls of Ortolan bunting, and it was done in succession with other calls that we know for sure are Ortolan bunting, and it's not associated with any other species. So, and it's more precise. It's also a call that is emitted during the day. It was just never found during the night. Yeah.
So thanks, super cool. I'm just very curious, like on who are the people who volunteer? Are they used to like naturalistic stuff and they just love that and continue for their free time, or like, and how do you find them basically?
Yeah, that's a really good question. Actually, the program was based on an already pre-existing base of ornithologists that were already taking part in nocturnal migration monitoring. So we just put the program out there, and then they were interested to join a national scale program. So they just joined, and then we're doing publicity basically through naturalist networks, and they just join. But they have a solid knowledge of already nocturnal migration calls for most of them.
Thank you. I think I have time by PT, but just a quick question. I think the answer will not be quick, but we can discuss.
I'll
later.
do my best.
Yeah, because I think that you will have acoustic data, but I think that at a point you will try to link to abundance data, like to measure some flux, something like that, of migrating bird.
Yes.
And how do you think you will manage this? How to link nocturnal vocal activity to migrating flux or abundance or something like that?
I'm not sure I completely understand, but I think because we have a lot of sites all across France, and most people are monitoring like multiple nights during the whole year, I don't really see why it's a problem. Sorry.
For example, you see you have 3 calls, but maybe there is 20 birds, and there is 3 calls, and there is hundreds of birds, something like that. And do you think it's maybe not relevant to make the parallel or to find a way to make some connection?
Oh, okay. So I think I know what you mean. This is actually the first chapter of my PhD, to try and make the link between what we hear and what we're missing, and to is that connected to your question at all?
Yes, kind of.
Okay, we can talk about it later, I think.
Thank you.
We have time for 1 or 2 questions.
Hi, thank you. Do you know what do they use to record the sounds, the bird sounds? Are they awake at night and they record the birds, or do they have autonomous?
Oh yeah, sorry, I didn't mention it's passive acoustic monitoring. So yeah, they have passive recorders that they leave on the whole night. So they put it at night, and then it ends in the morning, and they usually can leave them for multiple nights. Yeah, they're not just whole nights outside of their home. Some could, I think, but yeah.
Okay, so thank you